So it's the top of the hour. Good day, everybody, and welcome to the January 2018 NISS-Merck meetup. We have an excellent program today of speakers and discussants, but before we get to that, we need to do a little housekeeping. So I plan to do that in the next few minutes. All right, first, hopefully you all have good connections. This is a meetup, even though it's virtual, so we do encourage participation from the attendees. Unfortunately, because of the numbers, we have to mute the attendees' lines, but there are ways for you to participate. In particular, if you want to ask a question of the panelists, you should go into the chat function and select all panelists. The chat function is up in the gray band at the top of the WebEx Event Center. Select all panelists, type in your question, hit submit, and we'll do our best to get that to the panelists during the discussion time. Also, we do encourage communication between all the attendees. Again, you can do that in the same way. Go to the chat, but instead of picking all panelists, pick all participants. And that will send your comments out to everybody who is on the meeting site. And again, we encourage that. It's great to have people chatting along. My millennial sons say that this is the way the world should be today. So some of you younger folks out there can get us old folks started. The only comment about that is please no rude or disrespectful comments. We obviously want to keep everything at a very high level here. The other thing I'm going to say is a little infomercial about NISS. Last time when we did this, we had Ray Bain, who's on the board at NISS, give a presentation about what NISS is. I just want to go over the highlights of that. First, NISS is a nonprofit organization that was formed in 1990. Its primary mission is research, so it's a national research institute. They accomplish that mission in a number of ways. One is they have a very active postdoc program. They also co-sponsor and coordinate conferences, workshops, and expert panels throughout the community. So you're participating in one today. It's an independent research organization. So they do their own research based on grants, mostly from the government. And the way that I mostly get involved in this, and companies and often institutions, is through the NISS Affiliates Program, where they help sponsor workshops and conferences and things like that in various areas. So if you want more information, I encourage you to go to the website, and you can see the address on the screen, www.niss.org. Today we have a great set of presentations. The topic is ICH E9R1, so it's the ICH guidance on Estimands and Sensitivity Analysis in clinical trials. So it's about sensitivity analysis and missing data. We do plan to record the session and post it at the NISS website. So if you miss part of it, or if you want to go back and re-listen to part of it, look for it there, I don't know, probably a few weeks after we get done. And I'll remind you that last year's meetup, which was on multiple endpoints in clinical trials, that recording is there right now. Okay. With that, the agenda for today is Craig Mallinckrodt from Lilly is going to talk about missing data, fundamental considerations, and a brief history of research. And then we'll have Rod Little from University of Michigan talk about missing data in clinical trials, the NRC report. Devin Mehrotra will be next and leading a panel discussion. So first he will give some of his thoughts on those topics, and then lead a panel discussion between himself and the rest of the presenters. And that's the portion where if you want to submit a question, we will try to get it answered for you. So with that, I'm going to pass the ball to our first presenter. And let me give an introduction first. We are honored to have Dr. Mallinckrodt He is a senior research fellow at Eli Lilly. He led the pharma expert team on missing data and currently leads the Drug Information Association Scientific Working Group on missing data. Dr. Mellencrot is a fellow of the American Statistical Association and recipient of the Royal Statistical Society's Award for Excellence in the Pharmaceutical Industry for his book titled A Practical Guide to the Prevention and Treatment of Missing Data. So with that, it's all yours, Craig, and thank you again for participating.
Okay. Well, thank you very much for the introduction, Dan, and thanks to everyone for your interest in today's presentation. I'm going to start out, if I can get the slides to advance. It doesn't look like I have that capability at present. There we go. It's just a little slow. I'm going to start with an overview of the problem of missing data. I'll look a bit at some of the history of the research into the topic, a bit of history on industry practice, and then take a brief look ahead to set the stage for some of the other speakers to follow. So if we look at randomized clinical trials and think about when things go good, we get complete data. In this situation, there's not much controversy about what the treatment effect would be. We've got complete data, means of 10 versus 6. It's easy to estimate these means, and the treatment effect, no ambiguity, is a difference in mean change of 4 points. But complete data almost never happens in the real world. We almost always have situations where patients drop out at different rates for different reasons at different time points. And so the missing data causes us problems here. We can wonder how it is we might estimate mean change from baseline to endpoint in data like this, but we might more fundamentally wonder, what should we be trying to estimate? What can we estimate from data such as these? So missing data can certainly cause us some problems. Most importantly, missing data undermines randomization, the linchpin of inferences in our confirmatory trials. We need to bear in mind, though, that the definition and meaning of missing value or missing data is situation dependent. For example, if we have lost a follow-up in a depression trial, we truly are missing information that we needed to understand how the drug worked for that patient. But if we have death from a heart attack in a trial designed to test interventions to prevent heart attacks, we will again have some incompleteness in the data, but we're not missing any information because we know that the treatment did not work for that patient. So let's take a brief look at some of the history of missing data research. Way back in the day, in the 1970s and 80s, ad hoc approaches dominated our analysis of incomplete data, perhaps motivated mostly by the need to create balanced data sets to make the computations required for the estimations a little simpler. So we had observed case, last, and baseline observation carried forward type approaches. In the 70s and 80s, we did see some evolution with the definition of missing data mechanisms, the now familiar taxonomy of missing completely at random, missing at random, and missing not at random. We also saw the development of some missing at random-based approaches to move beyond the simple and ad hoc approaches, initially looking at likelihood-based repeated measures and multiple imputation approaches, and subsequently some inverse probability weighting approaches. Some of the key publications we saw along the way involving maximum likelihood evolved over the decades of the 60s, 70s, and 80s. A key publication on multiple imputation that also laid out the missing data mechanisms by Rubin, and then later the inverse probability weighting publications by Robbins and colleagues. In and around the 1990s, we started to see the development of some missing not at random approaches, with the shared parameter model appearing in the late 80s, the pattern mixture model that Dr. Little helped to champion in the early 90s, the Diggle and Kenward selection model at about the same time. During this period, we saw some criticism, but still continued use of the simple and ad hoc approaches in our primary analysis in confirmatory clinical trials. So the research was advancing, but the practice had not. In the 2000s, we saw a proliferation of simulation studies comparing MAR methods with the simple and ad hoc methods. We had multiple independent investigations in simulated and real data by industry, academia, and regulatory agencies that all largely showed the benefits of some of the newer approaches over the simple and ad hoc approaches. We had growing awareness that those simple methods were not broadly conservative as once advertised. And so there was a growing debate about appropriate choices for the primary analysis. Along the way, we did develop a widespread awareness of the need for sensitivity analyses fostered by the ambiguity the missing data caused. In our present decade, we've seen the development and refinement of ideas surrounding estimands. We've seen the advent of some new analytic methods, some in the multiple imputation framework that have been used for sensitivity and primary analyses, and also some likelihood-based analogs to the newer multiple imputation-based approaches. And we've seen some other approaches such as the trimmed mean, so lots of new research, lots of exciting things going on. And we've seen some refocusing on the need to prevent missing data. So let's look at the evolution of industry practice. Pre-2000, our analyses tended to be those simple and ad hoc approaches. Even though our computing power had improved, these were still a holdover from that era when computing power gave us limited options. Post-randomization events that we now refer to as intercurrent events were dealt with implicitly by choices made about the data collection and the statistical analysis, rather than doing it the other way around where the analysis was driven by the scientific question. We had the ICH E9 guidance appear in 1998. And in that E9 guidance, it talked about the ITT principle that asserts that the effect of a treatment policy can best be assessed by evaluating on the basis of the intention to treat a subject. And certainly ITT has influenced the handling of missing data, especially in the first decade of this century. ITT, though, was intended, at least some who wrote that document, the E9 document, tell me that their perception was that ITT was intended to define analysis sets and wasn't so much intended as a means to address missing data on individual patients. The ICH E10 guidance states that the need for rescue is an endpoint. So there's a bit of a conflict between the E9 and E10 guidance. And certainly, even if E9 tells us what to do for treatment policies, we wonder what if the key scientific question is about the individually randomized treatments rather than treatment policies involving those treatments. In the first decade of this century, we saw that industry was strongly moving away from simple and ad hoc approaches, primarily towards MAR-based, but some other approaches as well. Missing data was a very important topic at conferences and in regulatory industry interactions. Most importantly, I believe it was July 2010, the NRC expert panel released its report. Dr. Little will tell us more about that later in our current webinar. That panel provided 18 recommendations that largely can be condensed into 3 key pillars: to set clear objectives and define causal estimands, maximize adherence, and have a sensible primary analysis supported by plausible sensitivity analyses. Some of the consequences of the NRC report are that estimands have become a very important topic, and justifiably so, because estimands link the objectives and the analysis. The definition and proper handling of missing data depend on the estimand. As a consequence of the NRC report, we've also seen a new spike in methodology research, particularly pertaining to sensitivity analyses, which are now becoming routine practice, giving us straightforward approaches to assess consequences of departures from MAR and a number of other assumptions as well. We've seen greater, but in my opinion, probably still not enough emphasis on the prevention of missing data, and we've seen the advent of the E9R1 addendum, the draft guidance, to help clarify some of the issues that remain. We've seen some publications to help operationalize the 18 recommendations in the NRC panel. A series of publications resulted in the evolution of this process chart, where when we're planning clinical trials, we begin with the objectives, which are informed by the decisions we need to make. Those objectives then drive the estimands, what is to be estimated, which further informs design, analysis, sensitivity analyses. And importantly, this can be an iterative process. We don't want analysis considerations, for example, to completely change our view on what estimands are important. But if we do an iterative process, we can at least factor in some design and analysis considerations in our choice of estimand. We've begun to recognize that different stakeholders make different decisions and have different perspectives, which can lead to different interest in different estimands. For example, regulators and payers make one decision, and that one decision can affect the treatment of many patients. Physicians make many decisions, each affecting the treatment of one patient at a time. Patients make one decision about their own treatment. Sponsors have to make business decisions, and our interests can also vary depending on whether it's an exploratory versus confirmatory versus post-approval trial, short-term versus long-term, symptomatic treatment versus disease-modifying, efficacy versus safety, inpatient versus outpatient, and on the list goes, implying that in many clinical trial situations, there'll be need to address multiple estimands from the same trial. In terms of defining estimands, we've seen an evolution in clarity on just how to do that, and the R1 addendum is providing further clarity on that, instructing us that to define an estimand, we need to clarify the population, the endpoint, and the summary measure. And these are things familiar to us from specifying traditional primary analyses, but the new part is how to account for and deal with intercurrent events. Those post-randomization events that may be related to treatment and outcome, such as discontinuation of the intervention or discontinuation of the study entirely, the addition of or switching to rescue medication, and or death. So if we think about some example data here, there's 6 groups of data. There's really more, but we'll focus on these 6, where black X's indicate that the patients are observed on their initially randomized treatment. The blue X's indicate being observed on rescue medication. The red X's indicate that they were observed but not taking any treatment. And the dots indicate not observed. So 6 basic groupings here. Group 1 was adherent to the initial medication throughout. Group 2 was adherent throughout, part of the time on initial medication, part of the time on rescue. Group 3 took the initial med for a while, then rescue, then discontinued all medication. Group 4 took the initial medication, did not take rescue, but was observed taking no medication. Group 5 took the initial med, then took rescue med, and then dropped out and has missing data. And Group 6 took the initial med, dropped out without taking rescue medication, and was not further observed. And let's see, get this, there we go. So the R1 addendum provides us 5 general ideas on how to deal with those intercurrent events we saw in the previous slide. There's the treatment policy approach, where we would simply ignore the intercurrent events, and we'd have to define, for example, whether or not the use of rescue medication was a relevant intercurrent event or not. It could be part of the intervention we're interested in studying. The composite approach, where we use a modified definition of the variable for the summary measure, with the intercurrent events becoming a component of that outcome. Hypothetical approaches, where, for example, we might wish to estimate what would have happened if patients had not stopped the medication or had not taken rescue medication. The principal strata approach is where we restrict interest to the population of subjects, the stratum on which intercurrent events would not have happened on either treatment, or the while on treatment approach, where values of the variable in those patients up to the time of the intercurrent event is of interest. So we can kind of condense these ideas and think about some general estimands that are relevant for these 5 methods of dealing with intercurrent events. And it starts with whether our scientific interest is in the initial treatment or in the treatment policy. So do we wish to compare treatment A versus B, or policy A versus policy B, such as treatment A plus rescue versus treatment B plus rescue? And then we can further think about, are we interested in efficacy, or what some have termed the de jure estimand, the benefit of the drug when it is taken as directed, and these would lead to hypothetical and principal stratification approaches for dealing with intercurrent events. Or we could be interested in the effectiveness or de facto style estimands to assess the benefit of the drug as actually taken, where conceptually we've got the estimand being a composite of efficacy and adherence, and we would generally think about composite and while on treatment approaches for those types of estimands. And so perhaps in the future, as we work through these new guidances and understand them better and gain experience applying them in general practice, we might see some sort of roadmap to help guide our thinking, where we look at our primary estimand that's driven by the objectives, and we understand whether that estimand is targeted at an initial treatment or a treatment policy, whether we're going to deal with the intercurrent events through the statistical method or model, such as the hypothetical and principal stratification approaches, or whether we'll deal with the intercurrent events by definition or assumption, such as in the composite or while on treatment approaches. We've got a lot of work to do. We've got a rich history of research in this topic, but I'm more convinced all the time that our research needs to continue. We've made great progress. So we might say that while our research on missing data is a work in progress, there has been much progress in our work, and I look forward to much more as well. So with that, I'll turn it over to our next speaker.
Thank you, Craig. That was just an incredibly lucid presentation of a lot of very complicated concepts, and certainly a long history of this issue. So next up, we have, we're lucky to have another giant in the field. Rod Little is the Richard D. Remington Distinguished University Professor of Biostatistics at the University of Michigan, where he also holds appointments in the Department of Statistics and the Institute for Social Research. He is an elected member of the International Statistical Institute, a fellow of the American Statistical Association, and the American Academy of Arts and Sciences, and also a member of the National Academy of Medicine. He chaired the NRC Committee on the Prevention and Treatment of Missing Data in Clinical Trials, and we're lucky to have him here today to speak to us. So let me unmute him and pass him the ball. Okay, it's all yours, Rod. I will go.
Thank you. Now, I'm still seeing Craig's slides, so I don't know why I get my slides.
Yeah, just give it a second, I think, to... Yeah, I did. Craig, can you try clicking at the top there on the tab 2 where it says 2 and then your name? Maybe that'll help you. Are you able to see them yet?
Oh, okay. I got it now, yes. Okay.
Very good.
Can other people see them?
Yes, we can see them.
All right, great. So thanks, everybody, for joining, and thanks, Craig, for a fantastic overview, made life easier for me, I think. So I'm going to talk mainly about the NRC report that Craig mentioned. I'll have some other comments to make. I'll give an example of a sensitivity analysis and also talk a bit about the importance of alternative estimands. So this is the charge of the NRC panel to prepare a report recommendations for US FDA's development of a guidance for clinical trials and on appropriate study designs and follow-up methods to reduce missing data and appropriate statistical methods to address missing data for analysis of results. The focus of the report was mainly on confirmatory phase 3 type randomized control trials, but I think some of the comments would also apply to other kinds of studies. Just as a brief overview, one of the issues, Craig mentioned this, was how you define missing data, and this is the panel's definition, which I agree with, that missing data are unrecorded values that, if recorded, would be meaningful for analysis. So outcomes that are not defined for some participants are not considered by the panel as missing data. So a missed clinical visit, for example, somebody who didn't show up for a clinic but would presumably have information if they did, is an example of missing data. Quality of life for people who die is not considered to be missing data. So if you're really interested in quality of life, I think that's an issue that really only arises for people who are alive. And then outcomes, if individuals who discontinue a drug, if they had not discontinued, well, it kind of depends on the setting as to whether that's a reasonable counterfactual or not. Then this is sort of my elevator speech on the NRC study with some key take-home messages. So missing data undermines randomization, the linchpin of inferences in confirmatory trials. And there was a big emphasis in the study on limiting missing data. Craig also mentioned this. So limiting missing data should be a major consideration when weighing alternative design options. Analysis methods come with unverifiable assumptions, and limiting these assumptions is crucial. I think in the past there's a little bit of a tendency to say, well, perhaps not to worry too much about missing data in these earlier phases and just assume the statistician is going to fix the problem. But we can't really fix this problem without making assumptions, and we want to try and avoid these assumptions in this context. Careful attention to avoiding missing data in trial conduct can greatly limit the scope of the problem. And I think a lot of companies are paying more attention to this now, and perhaps I'm a little bit more optimistic than Craig is, but I think avoiding missing data is a much higher priority these days. Analysis methods need to be driven by plausible scientific assumptions, and then sensitivity analysis to assess robustness to alternative analysis models are needed. And the lack of a robust treatment effect across different choices of models really is not something the the sponsors want. So I think this kind of reinforces the idea of not having too much missing data to start with, because if you don't have a lot of missing data, then presumably the results are more likely to be robust. So design issues are paramount, and certainly on the topic of this webinar. The Estimand, the summary outcome measure of interest defined for the population under study, is the key starting point for the design of the trial. And alternative choices of Estimand can have important implications for trial design and implementation, and on the amount of missing data, and I'll give you an example of this later. Limiting missing data, therefore, should be a consideration when choosing an Estimand. I think there's a lot of scope for reducing the amount of missing data in this design by this kind of a strategy. One Estimand that I like that limits missing data is what I call an on-treatment summary. Craig used the term while on treatment, which is the same idea. So what is an on-treatment summary? A measure of the effectiveness of a treatment that only uses information while individuals are on the assigned treatment. Some examples: dropout as failure. Just define a dropout as a failure. Area under a curve measured in some way while on treatment. Dropout might be penalized in the sense that the area is then restricted to the time while on assigned treatment. Change from baseline to the minimum of dropout and end of the study, which is similar to LOCF, but really is defining an estimand to avoid the assumption of LOCF that there's no change after dropout. And then impute zero change for dropouts, which is similar to BOCF. So these are all suggestions for defining an estimand in a way that the cases that drop out, you don't have to try and impute what's happening after dropout, which generally involves a lot of assumptions. I think this kind of an approach could be used more in certain types of studies. Doesn't work for some things like if survival is the outcome, we can't really do this, but pain studies and other areas of clinical trials, I think this could be used more. Then there's various broader design strategies outside the estimand to reduce the occurrence of missingness. So run-in periods before randomization to identify who can tolerate or respond to the study treatment, having flexible dose titration studies to avoid people dropping out because of side effects, restrict trials to a target population for whom the treatment's indicated, reduce the length of follow-up period, which clearly has a downside to it, but generally reduces the amount of missing data, allowing rescue medications, as Craig was talking about, and then defining outcomes that can be ascertained in a high proportion of participants. And a bunch of other things in the report, which I don't have time to go into here, but none of these things come without some cost, so the benefits of these options need to be weighed against the costs. But missing data should, I think, be a consideration in the design phase. And then a bunch of strategies to limit missing data during the conduct of the trial. These tend to be fairly nitty-gritty, kind of, in a sense, perhaps obvious things, but not everybody's been following them as they could. So limit participant burden, set maximum acceptable rates of missing data and monitor the rates, provide incentives for investigators and for participants to stay in the trial subject to ethical guidelines, continual update of contact information, and education of the study staff on the importance of limiting missing data. So then, talking a bit more about analysis now, we had a set of principles. So we define what we mean by missing data in the earlier slide, formulate the analysis for inference about an appropriate and well-defined causal estimand, document to the degree possible the reasons for missing data and incorporate into the analysis. Some may be missing at random, others may not be missing at random. Decide on a defensible set of primary assumptions about the mechanism and then conduct a statistically valid analysis under those primary missing data assumptions. Statistical valid in particular meaning that you're taking into account the uncertainty in missing data. So just single imputation, for example, wouldn't apply there because you're essentially making up data and therefore not getting the right standard error. So there are methods that give you statistically valid analysis under the assumptions. And then sensitivity analysis to assess robustness of the treatment effect that needs to be pre-specified. And people are worried about pre-specifying a sensitivity analysis, but you know, in my consulting work, I'm seeing this is happening more and more. So it is possible to pre-specify such an analysis, and often the sensitivity analysis it doesn't necessarily doom the success of the drug. It really depends on the setting. Some missing data methods. Craig mentioned this a little in his study, but there's in his review, but complete case analysis dropping the cases that have any missing values, single imputation methods like last observation carried forward, baseline observation carried forward, inverse probability weighted methods, and then likelihood-based methods like maximum likelihood or Bayes or multiple imputation. And I don't have my things working, but the panel generally likes the second two of those over the first two. So I think there's clearly debates between probability weighting as method of analysis versus more likelihood-based analyses, but the last two bullets I think preferable to the first two generally. And sensitivity analysis parameters for missing not at random models cannot be reliably estimated. There's a long literature on this. Identifiability of parameters requires structural assumptions that are often very questionable, particularly in this clinical trial setting. So the panel tended to favor doing a sensitivity analysis rather than trying to make assumptions that will allow you to fit a missing not at random model. In many but not all situations, it might be reasonable to choose missing at random as the primary model, but then look at deviations from missing at random to assess sensitivity, to assess plausible deviations from missing at random. For missing at not at random models, there are two broad classes of models. There are others, but two of them are, let's see, I see my, looks like, interesting. I'm not getting I can't get my PowerPoint to work. I can see the rest of the slide in my side here, but I'm not sure how to get it anyway. There are selection models and there are pattern mixture models. Selection models factor according to the distribution of Y, where Y is the outcome variable, times the distribution of R given Y, while R is the indicator for whether you're missing or not. Pattern mixture models factor the other way into Y given R, that is the distribution of Y within each pattern of missing data times the mixed by the probability of R. These are suggested generic approaches. And I think the pattern mixture model approach has a lot of advantages in this setting, mainly because it's very easy to explain to clinicians. This example of a sensitivity analysis uses a sort of pattern mixture factorization and does look at deviations from not missing at random, actually coarsened at random, because this was in a survival analysis setting. It's the ATLAS trial that was assessing rivaroxaban, Xarelto, more familiarly known for reducing this composite risk of cardiovascular death, myocardial infarction, or stroke. 15,000 odd patients were randomized into 3 treatment groups. The 2 dosages of rivaroxaban and a placebo. This was on top of aspirin, by the way. The primary analysis was using a cost proportional hazard model and showed a statistically significant reduction in the primary efficacy outcome, which was this composite of the possible outcomes. Hazard ratio in favor of the treatment, combined treatment, was 0.84 with a confidence interval 0.74 to 0.96. There were concerns in the advisory committee about dropouts, so 5 to 10% of the cases didn't get to the final endpoint, and there was concern that of what I would call differential informative censoring. So informative censoring, or not coarsened at random, in the sense that the cases that dropped out had different outcomes than the cases that did dropped out. And if that was didn't cancel out, in some senses, if that's different in the treatment and the control, then you can get bias. So Janssen, the company that was the sponsor here, did a sensitivity analysis to try and address these concerns, looking at deviations from non-informative censoring. Actually, I prefer not coarsened, not at random, actually, is the term I like better. It eventually got into the paper. The interesting thing about this study is that there were 2 alternative estimands. There was an intention to treat estimate, which included all events occurring up until the end of the study, and then a modified intention to treat, where cases who dropped out, we only counted events within 30 days after the last study treatment, arguing that the effect of the drug, there was no effect after that 30 days. The FDA accepted modified intention to treat but also wanted to look at the intention to treat analysis. The interesting thing about this is the modified intention to treat has much less missing data than the intention to treat. So this is an example how the choice of estimand can have a big impact on the amount of missing data. The overview, don't have time to go into details here, but essentially you have a set of information up to the time of dropout. You estimate a hazard for the dropout at the time of dropout under non-informative or coarsened at random, but then differentially increase the hazard of an event in the outcome, in the rivaroxaban, the treatment group, by some amount, but only in the rivaroxaban treatment group, but leave the hazard in the other group as under coarsened at random. Multiply impute events between dropouts using multiple imputation. This was assumed a parametric Weibull distribution. You can't use a Cox model for this purpose. You need a parametric model. Combine results using multiple imputation combining rule, and then the tipping point being how much do you have to increase the hazard in the rivaroxaban group before you lose significance. So briefly, this is the sort of plot that you have. So it's looking at the hazard ratio. So 1 consists of losing significance. This is a confidence interval. This is the 0% here is the coarsened at random or non-informative centering analysis, the standard Cox model, and then increasing the rate of badness in the rivaroxaban group by a certain percentage until you get to the tipping point, which is where the confidence interval just crosses 1. Here, this is a very large inflation rate. This is for the modified intention to treat analysis where there's very little missing data. So things tend to be pretty robust to deviation from missing at random. For the intention to treat analysis, you get a much lower tipping point, 170%, because there's a lot more missing data, so things are much more sensitive. So this illustrates the effect of missing data on the choice of estimand on the amount of missing data and on the resulting sensitivity analysis. So trying to, if you're a sponsor, trying to get FDA to approve, or the agency to approve an estimand that limits the amount of missing data, I think is something to try. And I think FDA are more receptive to that than the past, where they have tended to had a very rigid sort of criteria about what the estimand should be. So to summarize, sensitivity analysis is a scientific way of reflecting uncertainty arising from potentially missing, not at random missing data, deciding how to implement and interpret the sensitivity analysis in the regulatory setting is challenging, I agree, but I think we are making progress, and I think Craig says a similar thing. The need and importance of the sensitivity analysis increases with the amount of missing data, which reinforces the idea of don't have missing data to start with. So that's where I'll end my discussion.
Thank you so much, Rod. A great presentation. The examples were certainly illuminating, and just a tremendous amount of material in a short amount of time, so we really appreciate that. The next part of the meetup will be simply a panel discussion. So Devin is going to start by giving some of his comments, and then ask a few questions of the presenters, and then this is a time when you, the attendees, if you have a question, please put it to all panelists in the chat, and we'll try to get to as many of those as we can. So, Devin, you're up.
Thanks very much, Dan, and thanks to both Craig and Rod for an excellent set of presentations. So I'm just going to give you just a few comments and then open it up for discussion. And perhaps the most important comment I'd like to begin with is to to get to a perception or a misunderstanding that the ICH E9 R1 amendment is about missing data. It's true that the term missing data often comes up in discussions about this addendum. But please understand that this addendum is not about missing data. And just by way of why this amendment was required to begin with. So back in 2014, towards the end of 2014, it was explained by regulators in Europe and in the United States and in Japan that it was their reflection that as they did their reviews of submissions from sponsors, that they found that it appeared that in many but not all cases, the analytical approaches that were being reported in these submissions were implicitly defining the treatment effect being estimated. And really what they picked up on in many cases was this lack of alignment between the objectives and the reported quantification of what was referred to as the treatment effect. And so really what that led to was a call sort of building on the work that the NRC, the excellent NRC panel report had begun the discussion. And this was to provide further clarity and to build on that. And ultimately it was all about defining some principles that people ought to be following in terms of ultimately what it is that you wish to estimate before you get to how you estimate it. What is the question of interest? What is the clinical question of interest? And so some of the examples that were sort of mentioned in the concept paper and then certainly early on in the discussions as we began to meet as a group were things like, there was lack of clarity in terms of the treatment effect. Is the intention to quantify the treatment effect that is due to the assigned treatment, regardless of other interventions such as rescue medication and so on in all patients, or is it the effect of the assigned treatment without rescue medication in all patients, or is it the effect of the assigned treatment only in the subpopulation of patients who can tolerate the assigned treatment? And so all these things are very important, and again, what the regulators were reporting to us as a group, that in their reviews of these submissions, that there was lack of clarity and there's lack of alignment. And furthermore, when it came to the so-called sensitivity analyses, again, there was a mixture of understanding, if you will, of what exactly is a sensitivity analysis. And the terms that were being used interchangeably were sensitivity analysis, supportive analyses, secondary analyses, and so on. And so really what that led to was a need to articulate in clear terms guidance in terms of a framework that people should be following. And indeed, that is what is the main feature of the ICH E9 draft guidance that has been posted for public comment, both in the United States, in Europe, in Japan, in Brazil, and so on in other regions. And really the framework that is defined sort of seems very natural and seems perhaps obvious to most, but because it was not being followed, there was value in explicitly clarifying what this framework is. And it really starts out by saying, as Craig nicely summarized, but so very briefly, what is the primary objective? What is the clinical question of interest? And that then, in a natural way, will lead to the estimand. And the estimand, of course, will then inform on what is the trial design. Once you've done that, then ultimately you are led to the method of estimation. What is your primary method of estimation? And in defining the estimators for the primary method of estimation, inevitably some assumptions are going to be invoked, and then the sensitivity analysis will then pressure test those assumptions. And importantly, the sensitivity analysis should be specific for a given estimand, because current practice, often but not always, is that different analyses are sort of thrown in together, and there's a counting game that is reported. Look, we have 1 out of 7 out of 9 analyses, and that is not really a principal way to do a sensitivity analysis. So a sensitivity analysis has a certain definition, has a certain discipline that needs to be followed, and these are some of the things that are articulated in the draft addendum that's in the public review phase right now. So with that, I thought I'll just say these few words just to lay out and to sort out perhaps some misperceptions about what the estimand is and why it was necessary. Indeed, we have begun to look as a group. This is the ICH-E9 group, and I represent pharma on that group, and we have begun to take a look at some of the comments. The group will be meeting face-to-face in Japan later this year, and we expect, probably at least 1,500 to 2,000 comments, borrowing from the kind of feedback that other similar addendums and similar guidance documents, the magnitude and the flavor of comments that have come in, for example, for E17 and so on. So at this point, we have 10 minutes left, and what I'd like to do is to begin the panel discussion, and I'd like to throw out maybe just one question for consideration by both Craig and Rod, but certainly invite others who are listening in to please submit your questions in the Q&A panel, and then I will take a look at those as best as I can, and then feed that into the discussion. So the first question I'd like to start out with, maybe Rod first, and then Craig, if you don't mind. And so there's a lot of discussion about using the missing at random assumption as a starting point in the context of estimation. And again, sort of the addendum, of course, talks about, be careful about what the estimand is, and then in the context of estimation, does MAR make sense? And at least as a group thus far, we have come to the conclusion that there are many situations where even as a starting point, the MAR assumption by itself may not be tenable, may not be reasonable. And therefore, current practice seems to be where you start out with the MAR as an assumption for the primary analysis, and then you pressure test that, and you see if you were to deviate from that, at what point do you tip? And so I guess my question here is, in those situations where even as a starting point, if the MAR assumption may not be tenable, what is a natural starting point, and what should one do in terms of sensitivity? It is a challenging question, and therefore, because the answer is not obvious, perhaps I can start with you, Rod, and then go on to Craig to see your thoughts.
Yes, okay. So one setting where I think missing at random is not a good starting point might be smoking cessation trials, where the assumption often in that area is if you have treatments to try and get people to stop smoking, then the people that don't show up at the end are probably not showing up because they've started smoking again. So that's sort of imputing a failure to the cases that drop out. So that's a simple form of, in a sense, on treatment summary. It's a missing not at random assumption for the cases that drop out. So that people do that in smoking cessation trials, it becomes a binary outcome, which is losing some information, I think, perhaps. If you are doing a sensitivity analysis based on that, then you might replace the assumption that everybody's that drops out is a failure by, there's some probability of the dropout being a failure and or starting smoking again, and then assess sensitivity of that probability. Where making the probability the same as what you're estimating under the other group would then be missing at random. So you're then moving from an extreme missing not at random assumption towards missing at random.
Thanks so much. Craig, any thoughts?
Yeah, a couple things, Devin. Thank you very much for the opportunity. I'd like to approach the question from two standpoints. First, in the new nomenclature of the R1 addendum, there are certain approaches to dealing with intercurrent events that will yield no missing data, and so the whole context of MAR becomes irrelevant because we're defining an outcome and an estimand in such a manner that there is no missing data, and the question becomes irrelevant. So here I'm thinking in particular about the composite approaches, the while-on treatment approaches, and to some degree also the principal stratification approaches. So then if we limit our discussion to the approaches where MAR is relevant, such as the treatment policy approach or some of the hypothetical approaches, the most common one being the effect if the patient had not stopped or switched, then within that context, as Rod was indicating, when MAR is not plausible, well, when is it the most plausible? MAR is defined conditional on covariates and previous observed outcomes. So in a repeated measures setting, especially with continuous outcomes where you can see gradients of change over time, MAR has a greater likelihood of being valid than in those situations such as Rod pointed out with smoking cessation, where the patient history might indicate a favorable outcome, but then all of a sudden the next observation which is missing could be completely different from the patient history prior to that. Those would be the situations, as Rod pointed out, where MAR is the least tenable, but when you tend to see gradual evolutions in observed values and patient histories over time, MAR is more plausible, but I applaud the ICH group for making it clear that there are some situations where we don't even need to worry about this, and we have some alternatives to having to assume MAR in the first place.
Yeah, great. Thanks, Greg. I was looking at my screen to see if any questions had come in, and I don't see any that have come in. Dan, am I looking at the wrong screen?
Yeah, there are a number of questions, so I can start. Here's one. In a time-to-event analysis, does informative censoring always mean MNAR, for instance?
Let me address that. So there's missing at random, and there's the term I like is coarsened at random. Coarsened at random being essentially random censoring. Censoring technically is not a missing at random assumption. If you look at my book with Ruben, we have a discussion of this. Other than that, I think we don't have time to go into more detail. But they're somewhat related. But for censoring settings, coarsened at random is sort of like missing at random.
Yes, thanks. This is Devin again. So one question that has come up, now I can see the questions several times, has to do with the principal stratification. And this was discussed at length by the E9 group. And really it spoke about in those situations where the effect of interest is in those patients who can tolerate the drug. And we were reporting on our own experiences where clinicians simply want to know if my patient can tolerate this drug, what will their efficacy look like? And so for that type of question, some implications or advice on design and analysis. Again, Rod, back to you.
Well, I mean, the Angres, Estimands, and Rubin paper in JASO is probably the earliest version of that issue, and I would. It's too hard, too extensive an issue to get into in a short amount of time. But maybe I should mention the Angres, Estimands, and Rubin paper is a useful paper. The principal stratification paper by
Rubin and Frangakis.
Frangakis and Rubin, yes, in biometrics is a very key paper in that area, and the instrumental variable methods they generally don't come cheap. I think doing principal stratification stuff does require making assumptions. I don't think you can get by them, and I think one of the main virtues of that framework is it makes clear what the assumptions are, which tend to be buried in other formulations. But that's about all I want to say about that, given the amount of time. So.
Thanks. So, Dan, I will hand it to you in 10 seconds, but before I do that, I just wanted to mention again that the E9R1 group did discuss this, and we just wanted to sort of point out that what is often done is that people will do a completers analysis or look at only those subjects or patients who tolerated the treatment, and then presume that is estimating what some people want to estimate in terms of the principal stratification strategy, but that doesn't quite do it. So again, in a short time, it's hard to address these, but let me just hand it back to you to close this meeting.
Thank you, Devin. I want to again thank so much the speakers and Devin for leading the panel discussion. It's a highly distinguished group, all three ASA fellows and highly distinguished in terms of accomplishments in this area. So I want to thank all three of you. I also want to give a special thanks to Christy Trang-Stein, who organized the session, and Kara Hackman for technical support. As I mentioned before, this session was recorded, and so in a few weeks you should be able to find it on the NISS website. And also, if you would, when you get out of the meetup, there's a short survey. If you would fill that out, there's only 3 questions, basically asking what you liked, what you didn't like, excuse me, and ideas for future meetups. So again, I want to thank everybody who organized it and participated. I want to thank the attendees for making it a large group, and we'll look forward to seeing you next time. Signing off.